today, so I'm a postdoc in the lab of Sergei Klizimsky, and I'm going to present some work that I've done.
Down. You can wait just till this bolus comes, and then we'll start the clock.
I'm not able to move.
I don't see my guy.
It's the up and down.
Yes, it's not. Not quite a few like this. Now
Okay, so I'd like to start by motivating the main problem of the talk. There's this idea in evolutionary biology that the same gene can impact multiple traits or can have effects on multiple traits. And you can also extend this to multiple environments. Same genes have multiple effects have different effects in multiple environments. And so this is known as pleiotropy. For the purposes of today's talk, we are going to limit ourselves to talking about this fundamental biological question: how does fitness of a population adapting to one environment change in a different environment? And you can think of a biological example, which is the one that I show here. Suppose we have a bacterial population, which is treated with an antibiotic 1. And this bacterial population develops resistance to this antibiotic. So it adapts to this antibiotic. And so the question is, would it also be resistant to a second, yet unknown antibiotic? Or would it also be, or would it lose its fitness? Or would it be more sensitive to the second antibiotic? And to start answering this question, we can try to look at it a little more conceptually on this plot right here. So on the x-axis, we have fitness in the home environment. So I'm defining here the home environment as the environment in which adaptation is happening. And on the y-axis, we have the fitness in a non-home environment. So in our previous example, the home environment was antibiotic 1, and the non-home environment is antibiotic 2. So you can see that I've just separated these into the axes of adaptation and pleiotropy. And there are 3 possibilities when it comes to pleiotropic trajectories. One is that there could be no change in the fitness in the non-home environment when there's adaptation to the home environment. And as you can see, you're going from left to right, so fitness is increasing. That's what we mean by adaptation. And we could have a pleiotropic fitness gain or a pleiotropic fitness loss. And the question is, can we really say what's going to happen? And we can, if we know how mutations change fitness in both environments. So we need to know the fitness effects of mutations in both environments, the home and non-home environments. And so our lab has come up with this kind of framework to understand this, which is known as the joint distribution of fitness effects, or the JDFE. And here on the x-axis, now I have the fitness effect in the home environment. And on the y-axis, I have the fitness effect in the non-home environment. And these bold lines show us the neutral mutations in both of these environments. And then we divide this kind of XY plane into beneficial mutations in both environments, deleterious in both environments, beneficial in one, deleterious in the other. So, and now if we go take a genotype and measure mutations and how these change fitness in these different environments, you'll get something that looks like this. And this is an example of a JDFE, or a joint distribution of fitness effects, in which mutations that are primarily beneficial in the home environment are also primarily beneficial in the non-home environment. And this can be used to predict for a given genotype pleiotropic trajectories. And you have something that looks like this, where we have where we only focus on the beneficial mutations, and beneficial mutations in the home environment are on an average also beneficial in the non-home environment. So we would expect fitness to simultaneously increase in both the home and non-home environments. And so this narrows down the questions that I'll be addressing during this talk. The first question is, how do the empirical joint distribution of fitness effects vary across environments and genotypes, and how the statistical structure of joint distribution of fitness effects constrain pleiotropic trajectories. And to start answering this question, first, I'm going to present this data set, which was collected in the lab and it's published by the lab. I was not involved in this project, but in this data set, they measured the fitness effects of around 100 mutations in a panel of 42 strains of yeast in 6 different environments. And you can see our environments right here, where we have the different temperature and pH combinations. And I just showed 3 representative strains, but as you can see, there's much more number of strains. And so now we have our environments, we have our strains and also mutations, so we can visualize these joint distribution of fitness effects and how they are supposed to look experimentally. And right here, to construct these joint distribution of fitness effects, on the y-axis, we have fitness effects on the non-home environment and fitness effect in the home environment on the x-axis. For the purposes of this talk, so this choice is arbitrary in this case. So because we are not really, there aren't really experiments on these pleiotropic trajectories. So for now, this choice is quite arbitrary. And we can start visualizing these joint distribution of fitness effects. So these mutations in different strains and environments. And these vary across the strains. So these shapes are quite different across strains. And they're also different across environments. So these JDFEs, the shape of joint distribution of fitness effects, or JDFE, vary across strains and environmental pairs. And so then we need to try to understand maybe if there's some structure to this variation in these JDFEs across environments and strains. And so to do this, I'm going to summarize very quickly the results that were found in a previous study about the statistical structure of these fitness effects in this particular experiment. So again, now we have a slightly different plot. So on the x-axis, we have fitness in the home environment. This can be fitness in any other environment. And we also, on the y-axis, we have the fitness effect in the home environment. So and these diagrams kind of represent the same mutation in different strains with different fitness in home environment. So as you can see, a mutation which on an average has a genotype or a strain which has a high fitness in the home environment, on an average is beneficial in the home environment, and a mutation which is in a background with high fitness is deleterious in a non-home environment. So we have this for only one mutation, and I'm going to ignore some of this noise for now, where we have these mutation and genotype specific effects, and just treat this as a line. I'm happy to take questions about the structure of the noise as well. So this is just one mutation, but as we saw, there were a lot of different mutations, and so we can represent these different mutations as follows. So these are the fitness effects for different mutations in the home environment as a function of the fitness in the home environment. And so these fitness effects on an average decrease. They go from being primarily beneficial to primarily deleterious as fitness increases in the home environment. And this is known as global epistasis. And I'm going to now also introduce a second observation from these studies, which is that all of these mutations cross this line of neutral mutations at around the same point, which is known as the pivot fitness. And from now on, I'm going to take fitness relative to this point, where mutations go from being primarily beneficial to primarily deleterious. So this is the line where mutations are on an average neutral in this setup. Okay, so moving on, we can now just write this down as a linear equation where we have the fitness effect in any environment. So delta F mge for a given genotype of a certain fitness decreases with a slope Bm, which is mutation specific. This is a function of the fitness relative to the pivot. So this is kind of the summary of these data. And the second thing to note, which is that now we are talking about we need to go from talking about one environment to talking about multiple environments. And so to do this, I need to introduce a second thing, which are these environment specific effects. So these average lines, they don't really all pass through zero. There's some variation around this pivot fitness, right? So we do this, we include this by adding this intercept term, which is an environment specific effect. And so if you look at, this is just for one mutation. Now we look at the same mutation in another environment, and this environment specific effect differs between one environment to the other environment. However, the slope remains the same in this specific data set, but I'm happy to discuss what happens if you kind of relax this specific assumption. And now if we look at all the, if you bring back all the mutations that we already had, we see that we first make a few assumptions about the structure of this eta term, and we assume that they are normally distributed and uncorrelated, and the environment specific effects are uncorrelated. So as you can see right here, I'm just going to point this out. So this mutation here does not land at the they're kind of jumbled across space. So the idea is that they're uncorrelated. These points here, these indicated in triangles, are uncorrelated between environments. And this holds for cross temperature pairs in the in this specific experiment, but I'm also happy to discuss what happens if there are some correlations. Okay, and we don't need to assume any specific parametric form for the slope distribution, but we need to assume that the slopes are mostly negative, and this is okay for all environments. Forget this S, I added that very last minute. Okay, so now we are ready to construct the joint distribution of fitness effects from the global epistasis equation that we already saw. Now we are back to our old plot, where we have the fitness in the non-home environment as a function of the fitness in the home environment, right? So these are the x-axes here. And I also indicate this point here, the pivot fitness in the home and non-home environment on this 2D plot. And so we can start constructing these joint distribution of fitness effects. And let's start by focusing on a genotype here. Suppose we have a genotype which has low fitness in both the home and non-home environment. And I plot the fitness effects, delta F and H, as a function of delta F H. And so what is this supposed to look like? Now we can see on the plot to the right that mutations that are primarily beneficial in the home environment are also primarily beneficial in the non-home environment. So what we would get is something like this, right? So we would get mutations that are simultaneously beneficial in both environments. Similarly, I'm going to focus on a second genotype here, which I indicate in pink. And so this has a low fitness in the home environment and a high fitness in the non-home environment. So mutations that are primarily beneficial in the home environment are primarily deleterious in the non-home environment. So we're just taking this and kind of putting it right here. And now we can do this for all parts of this fitness and non-home environment versus fitness and home environment space, and we get something that looks like this. So we can actually write down these, assuming, as I said, that the etas are normally distributed, and assuming any kind of slope distribution, we can write down this joint distribution of fitness effects as a function of the fitness in the home environment, fitness in the non-home, and fitness of the non-home environment, and some more properties of this distribution. So we have this distribution, these joint distribution of fitness effects. And the main thing to note here is that these JDFEs depend on the strain fitness relative to the pivot point. So they depend on the fitness in the home environment and the fitness in the non-home environment. And so to move on from here, we come to the second question, right? So how does the statistical of JDFE variation, which was discovered in this experiment, constrain pleiotropic trajectories? And to begin to answer this question, we can now use these JDFEs here, which we constructed before. And now that I bring back the theory that was developed in our lab, we see here, focusing on this blue genotype, that mutations that are primarily that are on a beneficial mutations are on an average also beneficial in the non-home environment. So beneficial mutations in the home environment are also primarily beneficial, are on an average beneficial in the non-home environment. So from here, and similarly, we can do this for the pink genotype, and we see that mutations that are beneficial in the home environment are on an average deleterious in the non-home environment. And so from this, you can calculate these statistics which predict these trajectories, and we get something like this, right? So we expect the that if there's a genotype which is simultaneously has low fitness in the home and non-home environments, we expect the fitness to we expect pleiotropic fitness gain, and if there's a genotype which has low fitness in the home environment and high fitness in the non-home environment, we expect a pleiotropic fitness loss. And we can actually show that as long as we can actually show that only thing that matters is this relative position of this genotype with respect to the pivot. And so how do these trajectories actually look like? Well, it turns out that this pivot fitness right here, so in the non-home environment, I'm talking about the pivot fitness in the non-home environment, is actually an evolutionary attractor. So all the trajectories that start anywhere in this fitness and home environment and non-home environment space, they all converge to the pivot fitness. And so they converge to the point where mutations are on an average neutral in the non-home environment. But you can note, just to make it kind of easier to see on this plot now, if you just focus on the home environment, you have adaptation, right? So what happens is that you kind of ride along the wave of beneficial mutations, so you get something that looks like this. You have a fitness trajectory that just goes along all of these beneficial mutations. However, in the non-home environment, what matters is the average mutation, given that it's beneficial in the home environment. And so what we see is that while we have adaptation here, we can have a pleiotropic fitness gain or a pleiotropic fitness loss, which just depends on the starting fitness point of the genotype in the non-home environment. So the only thing that matters in this setup is where the fitness starts in the non-home environment, and they all converge to this point right here, which is the pivot fitness, where mutations are on an average neutral. Okay, so I'm just going to summarize this work and give you the take-home messages. So what I'd like you to remember, the first thing is that the empirical joint distribution of fitness effects from the experiments vary greatly across strains and environments. So they do depend on the genetic context and the environmental context into which, so mutations depend on both the fitness effects of mutations depend both on the genetic and environmental context in this specific experiment. The second thing that I'd like you to take away is that global epistasis, so this dependence of mutations, this very specific type of dependence of mutation effects, the fitness effects of mutations on the background fitness, this constrains the joint distribution to depend also on strain fitness. And the last thing that I'd like you to take away is this pleiotropic trajectories are predicted to converge to an attractor in this setup. So with this, I'm happy to take any questions, answer any more details of any of these calculations, and so on. Yeah.
Oh, that's a great talk. Thank you. Are there conditions under which you might expect the pleiotropic trajectories to converge to say different multiple attractors, or fails to converge at all?
Yeah. So one obvious thing here, I think you can already see it here, right? So I started with this assumption that we have uncorrelated pivot points, right? So these points here are uncorrelated. So in this case, the typical mutation over the typically beneficial mutation here will on an average be neutral in the second environment. But you can just imagine that you have a perfect correlation between these points, right? So if you have perfect correlations between these points, then what happens in the non-home environment is exactly what happens in the home environment. So as soon as you start having correlations between these points, where this line crosses the neutral, so this point, the pivot point, right? So as long as you have variation, have covariation between this noise right here, then you can have trajectories that do not converge to this specific point. But so what this kind of tells us is that these, if we have this uncorrelated structure, which we do see in the data, what this means is that the mutations that are typically, so you have two kind of, you can divide this into two spaces, right? You have mutations here that are primarily beneficial in both environments. This is a constraint imposed by global epistasis. And right here you have simultaneously deleterious mutations. But what happens right here is a little bit more special, because you have some mutations that are specifically beneficial and some mutations that are like non-specifically beneficial. And so these correlations kind of would change the pleiotropic outcome. But if environments are sufficiently different so that these specific mutations are also different, are uncorrelated, then yeah, this is what you expect. Yeah.
That's great. That's really nice. I'm wondering, you gave your example with antibiotic resistance, and there's all this literature on kind of collateral sensitivity, right?
Right.
I'm wondering whether have you been thinking about kind of taking this approach to actually really understand patterns of kind of collateral sensitivity and so forth?
Right now, no, but that's actually a good direction to take this work in. Yeah. Are there any more questions? Yeah.
I may have mistaken something, but I was a bit puzzled by the fact that it doesn't depend in how often you experience each type of environment.
Right. So maybe I should have been clearer in communicating this, but what we are seeing is that they're only experiencing one environment. They're only experiencing the home environment. And what's happening in the non-home environment, so it's kind of like, it's a setup where you first measure the fitness. Like, this is not the actual experiment, but an experimental setup corresponding to this would be you first measure fitness in the home and non-home environment. You then allow for the strain to adapt in the home environment. And then now you are measuring both in the home and non-home environment, right? So that's the setup. So these are not fluctuating or alternating or nothing. So these are just, yeah, two different environments.